Good evening to all the CUET aspirants. So, here we are in the concept of post-fertilization events, wherein today we are going to learn in detail about endosperm development, embryo development, types of embryo development both in dicot embryo and monocot embryo, followed by seed development, fruit development and some special concepts like apomixis and polyembryony. Let us get started. First, if you look into the post-fertilization events, we have 4 major post-fertilization events to be discussed in angiosperms. First is endosperm development. Endosperm development will precede the formation of embryo. Okay, even before the development of embryo, endosperm development will takes place. And post-fertilization, ovule will turn into seed and ovary wall will turn into fruit. Fine, right? How seed is formed and fruit is formed. These are the 4 major post-fertilization events that we are going to look into. First, if you look into the endosperm, endosperm in angiosperms whose ploidy is triploid, which is formed by the fusion of polar nuclei or central cell whose ploidy is diploid along with the male gamete whose ploidy is haploid, that has led to the formation of triploid primary endospermic nucleus. And this primary endospermic nucleus maturing to endosperm is on different pathways again. We will look into it. And the nature of endosperm will also vary in different plants. Here if you see in cereals like rice and wheat, the endosperm is starchy. You will see starchy endosperm in cereals like rice and wheat. In cereals like maize, you can also see proteinaceous endosperm, that is aileron layer over the starchy endosperm. If we have to take the rough diagram of, here is scutellum, rough diagram of corn kernel. If you see, this is scutellum which is papery cotyledon, and here will be the embryonal axis at the tip, which is the coleoptile which is the proteinaceous sheath which is covering out the plumule, and at the base you will have radicle and the sheath which is covering the radicle is coleorhiza. This is the papery cotyledon which is scutellum, above which you have the endosperm. At the center you will have starchy endosperm which is encoated by a protein rich layer called as aileron layer, which is proteinaceous endosperm. So, in maize seeds you will see proteinaceous layer also. Clear, right ma? And then castor and coconut. In castor and coconut you will see oily endosperm. Castor and coconut, oily endosperm. Ivory palm and date palm, if you observe, ivory palm will be having cellulosic endosperm, date palms will be having hemicellulosic endosperm. These are different compositions of endosperm in different seeds. And in few families like Orchidaceae, Podostemaceae and Trapezae, we are yet to discuss about families from taxonomy of angiosperms. We will do it, but as of now you remember that in these families the endosperm is absent: Orchidaceae, Podostemaceae and Trapezae. Followed by stony endosperm you will see in betel nut, that is areca nut, and in date palm, that is Phoenix dactylifera. In these you will see stony endosperm, followed by diploid endosperm, which is quite a times asked question. Even if you want to see in your CUET PYQs, you can check it out, that ploidy of endosperm is also a question. Usually it is triploid, but there is one exception, that is about Oinothera, which is having diploid endosperm. These are different examples of endosperm. Now you see we have 2 patterns of endosperm development. One is free nuclear endospermic development, free nuclear endosperm development, and cellular endosperm development. Free nuclear endospermic development and cellular endospermic development. Free nuclearic endospermic development, if you see, primary endospermic nucleus will undergo nuclear division, and there is no cytokinesis that is following up the karyokinesis. Immediately after karyokinesis, there is no cytokinesis that is taking place. So, complete nuclear division without cell wall formation will lead to the formation of a free nuclear endosperm, and cellularization will happen after complete nuclei formation. Cellularization will occur at the last or at the end. Whereas, if you see in the cellular endosperm, each karyokinesis is followed by cytokinesis, and you will see the cellular endospermic development like this without any free nuclei formation. Both free nuclear endospermic development and cellular endospermic development you will see in a single plant. I can give you the best example of coconut, where at the center the liquid endosperm which is present, liquid endosperm. This liquid endosperm is the result of free nuclear endospermic development and the thick endosperm which is formed, cellular endosperm which is formed. That white pulp which we have, that is the result of what? Cellular endospermic development. So, these are the two modes of endospermic developments. And then here you see fate of endosperm. We have n number of seeds. In few, endosperm is still persistent in the seed. In few, endosperm is absent. It will be completely utilized by the embryo development. In those seeds where endosperm is still persistent are known as albuminous seeds or endospermic seeds. They are known as albuminous or endospermic seeds. In those seeds where endosperm is completely consumed by embryo development, such seeds are known as exalbuminous seeds or non-endospermic seeds. Mostly non-endospermic or exalbuminous seeds will be of monocots. Mostly monocot seeds will be, mostly dicot seeds will be exalbuminous or non-endospermic. Ganesh, please repeat your doubt. I didn't get you exactly. Yes, you are right, Surya. Pea and beans are the examples of, no. P and B are the examples of exalbuminous seeds. Albuminous seeds mostly they will be monocots, monocot seeds. Fine, right. Only dicot example I can give you is castor for albuminous seeds. Here some monocot seeds like orchid seeds can also show the exalbuminous condition. Yes, you are right. Fine, right? This is about albuminous and exalbuminous seeds. I hope this is clear, right? And now, embryo development. So, you see here in embryo development, first we will discuss about dicot embryonic development and different types of dicot embryo development. Here first let us look into the zygote. Always we have to fix the position of zygote based on the micropylar and chelicil end. Here you see this is the micropylar end and this is the chelicil end of this zygote, okay, of let us consider orthotropous ovule. This is not anatropous ovule, this is a straight over, let us suppose, which is orthotropous ovule. And here zygote will have micropylar end and chelicil end. Clear, right? Here one zygote, which is a single cell, will undergo first transverse division and it divides into two cells. Towards the chelical end, terminal cell is formed, and towards the micropylar end, basal cell is formed. And here you see successive transverse divisions of basal cell and terminal cell. If you observe, the basal cell will continuously undergo division and it forms suspensor. Okay. Basal cell will undergo transverse divisions of 8 to 10 cells and it forms suspensor. The tip towards the micropylar end, the suspensor cells, the straight cells are known as suspensor. The uppermost cell of the suspensor, which is close to the micropylar end, is known as haustorial cell, which is close to the micropylar end, and that suspensor cell which is attached to the embryonal octant. Terminal cell, basal cell will again undergo transverse divisions forming suspensor. Terminal cell will give rise to embryonal octant. Embryonal octant. Okay. That suspensor cell which is connected to this embryonal octant is known as hypophysis. Remember, the suspensor cell is the resultant of the transverse divisions from the basal cell. The tip of the suspensor cell or the apical cell which is towards the micropylar end is known as haustorial cell, and the cell that is connecting the embryo is known as hypophysis. Clear, right? And then you see here the terminal cell undergoes division in all the planes, forming out epibasal cells and hypobasal cells. The lower tier is known as epibasal cell, upper tier will give rise to hypobasal cells, and this embryonal octant will directly give rise to the epibasal cell and hypobasal cells will give rise to the outermost region, like plumule, cotyledons and outermost epidermis. All these are formed, epicotyle, hypocotyle, these are formed from the epibasal hypobasal cells. Hypophysis, which is connecting these two cells, both suspensor and the embryonal octant, hypophysis will give rise to root cortex and root apex. Fine, right? This is how the octant embryo with suspensor and the haustorial cell will further divide, forming out a globular embryo. This forms out what? Globular embryo. And then you can see heart shaped embryo. At the center point, plumule is present, and this heart shape, these two projections or elongations will represent two cotyledons. And this will later give rise to root cortex and root apex. This is how the embryo development will take place. If you clearly look into the dicot embryo which is formed after the heart shape, first zygote undergoes a transverse division forming terminal and basal cell. Basal cell is towards the micropylar region, terminal cell is towards the chelical end. And here the one which is towards the basal cell which is towards the micropylar region undergoes sequential transverse divisions, where the apical cell is the haustorial cell and the base cell of the suspensor is hypophysis. And here terminal cell will undergo division in all the planes, giving rise to embryonal octant, which later gives rise to plumule cotyledons here, and hypophysis gives rise to root cortex and root apex. This leads to the formation of a globular embryo, and then cotyledon growth will lead to the formation of a heart shaped embryo, and then you can see a matured embryo with two cotyledons, and at the tip where the plumule is present, and at the base you can see the radicle. Base of the radicle you can have root cap, and here you can see hypocotyle. From hypocotyle only the radicle and root cap will arise. This is the complete description of dicot embryonic development. But here you will see different types based on whether suspensor is present or absent, or whether the embryonal octant is formed from terminal cell or basal cell. As such, you will have different types. Here we see first crucifer type or capsular type or classic dicot type, which is a classic dicot type. So, you see just now whatever we have learned, it is from it is of crucifer type only. Just now what type of embryo we have learned, that is crucifer type only, which is a classic dicot pattern. So, here for example, this type is observed in Capsula brusa pastoris and Brassicaceae family members mostly. Okay. Here you see first division is transverse division forming terminal cell and basal cell. This we have understood. Basal cell will be towards micropylar end, where this forms out a multicellular suspensor cell, and at the terminal cell this divides into planes where 2-celled forms out 4-celled condition. Later this leads to octant stage, further develops into globular embryo and heart shaped embryo. Do remember, heart shaped embryo is formed only in the dicot embryogeny. You will not find heart shaped embryo in monocot embryogeny. And here because of the periclinal divisions, protoderm is formed, which is the outer layer, and the inner cells which are present, they will lead to the formation of ground meristem and procambium. Characteristic features, embryo proper mainly forms what? Terminal cell. Suspensor is well developed here, and embryo shows globular embryo develops to heart shaped torpedo stage, which is a typical dicot sequence. So, this is about crucifer type, where basal cell forms the suspensor, terminal cell forms the embryonal octant stage. Remember this, these 2 points are very important with examples. Clear, right? This is about crucifer type, which is the main dicot type of embryogeny. And next type, if you see, solenoid type. In solenoid type, both terminal and basal cells will contribute in the formation of embryo. Both the cells will contribute. Examples you can observe it mainly in the Solanaceae members like Solanum, Datura and Nicotiana. First division often is transferred, same like that in dicots, but basal cell derivatives also contribute to embryo proper here. Okay, along with terminal cell, basal cell will also contribute in the embryo formation here. Suspensor is shorter and thicker than crucifer type. In crucifer type you can see 8 to 10 cell long suspensor cells, but then here it is short and thick in solenoid type. Early embryo becomes more robust. Basal contribution makes the lower embryo region comparatively larger. Lower embryo region will be comparatively larger. So, this is about solenoid type. And third type is caryophyllid type. Remember, many a times the questions are put on the types of embryogeny. Yeah, here we go with this. Examples, usually you will observe this in Caryophyllaceae members. Basal cell either does not divide, either it does not divide, or it forms only very small cell with minimal role. Okay, it forms a very small cell with minimal role. Suspensor is completely absent or extremely reduced here in caryophyllid type. Major point to be remembered and highlighted. Here suspensor cell is either absent or it is short-lived. Embryo develops almost entirely from terminal cell. Only from terminal cell the embryo is developed, and suspensor cell is completely reduced or absent in caryophyllid type. Okay. And then chenopodiad type. In this type, if you see examples, Chenopodium, Amaranthus, Chenopodiaceae. Okay, Amaranthus, Chenopodiaceae. Key steps here, if you see, suspensor is small and it is not long here again. Terminal cell divides and dominates the embryo formation. Terminal cell itself dominates the embryo formation. Basal derivatives may remain limited. Embryo becomes globular quickly. Okay, quickly the embryo becomes globular. Suspensor present, but it is very small. Embryo is mainly terminal cell derived. Okay, this is about Chenopodian type. Belongs to the family Chenopodiaceae or Amaranthaceae. Asterad type, wherein irregular early divisions happen in this type of embryogeny. and this is observed in Asteraceae members. Best example is Helianthus. Key steps if you observe, early divisions in embryo is proper are less regular than capsula. These are less regular than capsula. Suspensor generally is poorly developed suspensor. Embryo will become globular mass, it becomes a globular mass early, later it differentiates into organ. Irregular division pattern and reduced suspensor are the key features of this asterate type. Next, piperant type, which is primitive or irregular type of embryogeny or embryo sac formations, observed in Piperaceae family. Early divisions are irregular, suspensor is poorly developed, often it is treated as primitive embryogeny. Okay. The highlights of this pattern of embryogeny includes irregular embryogeny, reduced suspensor. Fine. So, this is about piperat type. Now, if you take a keynote, this single table is also enough with examples. Okay. If you can have a screenshot of this single table is also enough. Here you see first crucifer type, where is suspensor is long. In crucifer type you can see long suspensor whose contribution to the embryo, where terminal cell only is contributing to the embryo formation. Example includes Brassicaceae. Solenard type, it has short suspensor and contribution to the embryo, both basal and terminal cell will contribute. Example is Solanum. And Caryophylla type, suspensor is absent, contribution to embryo, terminal cell only. Example is Sagina. Asterate type, where reduced suspensor is present, mostly this contributes, mostly terminal cell contributes to the embryo development. Example is Helianthus type. Ctenopodia type, suspensor is small and this contributes, mostly terminal cell contributes to the embryo formation. Example is Ctenopodium. Piperite type, irregular suspensor, indistinct embryo, which is primitive type or type of dicot embryogeny. Example is Piper. Fine, right? If you want, take a screenshot of this diagram, very important. This table is very important. Single table covers all the types of embryogeny. Fine. And now you see monocot embryogeny. In monocot embryogeny, zygote first divides into two cells and then one of the cell will again undergo division. See, it divides into two cells, terminal cell and basal cell. Basal cell will again undergo division, forming out middle cell and suspensor. So, the apical cell will be suspensor, next will be middle cell, and at the base you can see terminal cell. Terminal cell is towards which end? Terminal cell is towards the chaliceal end and suspensor will be towards the micropylar end. Here the middle cell itself contributes in the formation of plumule, epicotyle, hypocotyle, radical and cotyledons. So, the major contribution is from middle cells. Here after continuous divisions you can see a single cotyledon and above which a plumule is formed. So, you can see a globular embryo. Here at the height towards the micropylar end, At the chalazial end you can see cotyledon, plumule, and towards the micropylar end you can see hypocotyle and radicle close to the suspensor end. Globular stage itself is the last stage of the monocot embryogeny. Here if you can see the rough diagram of maize embryo, wherein you can clearly make out this as what? Embryonal axis. The tip of the embryonal axis is the shoot apex. Covering the shoot apex you can clearly make out the sheet, that is coleoptile. Coleoptile is the protective sheet which is covering the shoot apex. Coleoptile is the protective sheet which is covering up the shoot apex. At the base radicle will be present, below the radicle root cap is there. Below the root cap you can see the protective sheath which is covering the radicle, that is coleorhiza. Both coleorhiza and coleoptile you will see only in the monocot embryo, remember that. And here there is only single cotyledon which is papery cotyledon, that is scutellum, which is papery cotyledon. Let us draw the structure of the complete seed. Here if you see, this is the endosperm, where the outer region is, this outer sheet is proteinaceous layer, that is aileron layer. At the center starchy endosperm will be there. This scutellum will draw out the nutrients from the endosperm and it will give to the embryonal axis, which will lead to the germination of the seed at the time of embryonal axis development. Clear till here? Understood everyone about monocot embryogeny? Now we will learn out what are different types of monocot embryogeny. First about the general plan of monocot types. Just now we have learnt monocot wherein one cotyledon, that is scutellum, embryo is lateral relative to the endosperm, coleoptile protects plumule, coleorhiza will protect the radicle, and suspensor often includes specialized hostorial region. This specialized hostorial region is aiding the absorption in monocot embryogeny, especially in some grasses. In some grasses, the remnants of secondary cotyledon are also present. The remnants of secondary cotyledon is known as epiblast. In some grasses, remnants of secondary cotyledon are present. The remnants of secondary cotyledon is known as epiblast. Clear till here? Any doubts anywhere ma? And then grass type, if you see in poaceae or maize type of embryogeny, what is it formed? Like zygote first divides into terminal and basal cell. Terminal is towards the chalazial end, basal towards the micropylar end. Basal derivatives form a suspensor, often with hostorial cell. This hostorial cell will penetrate to the endosperm. Fine. Terminal cell will form embryo proper, but development will become asymmetric here. Okay, asymmetric pattern of development is seen. A lateral outgrowth becomes what? Scutellum type of cotyledon. Shoot apex forms and becomes enclosed by the coleoptile. Root apex becomes enclosed by the coleorhiza. Okay, this is the diagnostic point. Formation of scutellum, coleoptile and coleorhiza. Fine, right? This is observed in poaceae members. Next type is alisma type. More like dicot. Okay, more like dicot embryogeny, wherein you will see this is in alismataceae, wherein early embryo development is regular, which resembles dicot patterns. Suspens are present but not strongly specialized like grasses. Cotyledon develops but not into grass like scutellum. Okay. What is the main point to be observed? Monocot embryo without strong grass specializations that you will see in alisma type. Mostly aquatic monocots you will observe this. Next is orchid type of monocot embryogeny, where tiny embryo is seen. Tiny embryo with little differentiation. Examples: orchids, Orchidaceae family. Embryo remains very small and undifferentiated embryo, very small and undifferentiated embryo. Endosperm is usually absent or poorly developed. Seed is dust-like, which relies on mycorrhiza, fungal associations for germination. Very minute embryo and there is no endosperm, and it is dependent, the germination of the embryo is dependent on mycorrhiza. Yeah. Next is Dioscorea lilium type tetrad. Lilium type trends commonly discussed in monocot variations observed in Lilium and Dioscorea, where embryo may be relatively larger than orchid. Embryo is larger than orchid size, suspensor varies and cotyledon develops into a leaf-like structure. This is the characteristic feature. Cotyledon develops into a leaf-like structure. Example, if you see monocot embryo with single cotyledon but not grass special organs are present. No special organs like you see in grass embryogeny are not present, and the key feature to be highlighted here is cotyledon type is like single leaf like structure. And here is the table, okay, where you will see features like cotyledons, 2 dicots are present and monocots are just 1. Early stages globular, turning to heart shaped and then torpedo stage. Protective sheets are absent, coleoptile, coleorhiza are present like in grass. Suspensor is often clear and may be long like in capsula, often with hostorial function. Embryo position, often it is embedded and central, and here in monocots it is often lateral in endospermic grains. Fine, right? These are different characteristic differences between dicot and monocot embryogeny. Now you see seeds. We have different types of seeds. First you have to know what are the main components of seeds. Seed has 3 main components. One is embryonal axis. Embryo consists of, first is seed coat. Seed coat, especially in dicots, you see outer seed coat is known as testa, inner seed coat is known as tegmen. Next, second region is cotyledons. Okay, cotyledons, they based on the number of cotyledons we have 2 types of seeds. We can have 2 cotyledons or a single cotyledon. Third is embryonal axis. Okay, third is embryonal axis. Embryonal axis will develop into both upper plumule and lower radicle. Here we see 2 different types of seeds, both dicot and monocot seed. Here you can see first dicot seed, for example bean. In few endosperm is also present. Based on the presence or absence of endosperm also we have 2 different types of seeds. First you see here in the seed type, outer region is testa and inner will be tegmen, and here epicotyle will be there followed by hypocotyle. Okay, and hypocotyle will give rise to radicle, epicotyle will give rise to plumule, and you see cotyledons are known as embryonic leaves which will give the enough nourishment at the time of seed germination. Okay, same likewise if you consider in the monocot, the embryonal axis, epicotyle, hypocotyle, plumule, radicle, coleorhiza, coleoptile are present. Papery cotyledon, that is single cotyledon is present and endosperm do exist. Whereas in dicot seed, endosperm will be absent mostly, they are exalbuminous seeds. This is about seed description and components of seed. So, mature seeds you can see endospermic, non-endospermic seeds. Even after seed formation, in whichever seeds endosperm is still present, those are known as perispermic seeds. Persistent endosperm even in the mature seeds is known as perispermic seeds. Clear, right? This is about albuminous, exalbuminous seeds. In the beginning only we have learnt. Now look into the types of fruits. We have different types of fruits. Ovule develops into seed, ovary develops into fruit. If only ovary is contributing in the fruit development, then those fruits are addressed as true fruits. Along with the ovary wall, any other part of the thalamus or any other floral part is contributing in the fruit formation, those fruits are addressed as false fruits. And fruits which are formed without fertilization are known as parthenocarpic fruits. First we will see true fruits. True fruits examples mango, kiwi, grapes, likewise we can take n number of fruits. False fruits, best example you can take is apple and strawberry, where along with the ovary wall thalamus is contributing in the fruit formation. Bananas and seedless grapes are the best example of parthenocarpic fruits, where fruit is formed without fertilization. Now let us look into the special modes of reproduction like apomixis. Apomixis is a concept or a phenomena which mimics the sexual reproduction. Okay. This gives rise to diploid seeds without fertilization. And this is by two types. One is known as adventive embryony or sporophytic budding. And second type is known as recurrent agamosperm. So first you see adventive embryony or sporophytic budding. What type it is? Look into it carefully, wherein any of the cells from integument or nucellus will differentiate into embryo directly. Okay. Nucellus or integuments from the ovule. Whose ploidy is diploid, right? So they will directly differentiate into megaspore. Megaspore which is diploid, when diploid nucellus or integuments are differentiating into a diploid megaspore, diploid megaspore will eventually give rise to a diploid embryo, right? Embryo sac, diploid embryo sac to diploid embryo. Likewise, through adventive embryogeny or sporophytic budding, diploid seeds are formed. At the end, the seed formation which is formed because of this is known as apomictic seed. Recurrent agamospermy, when here in sporophytic budding, what happens? Any of the sporophytic tissue like nucellus or the integument cells are undergoing budding and they are getting differentiated into embryo. But here megaspore mother cell in recurrent agamospermy, instead of undergoing meiotic division, here it undergoes mitotic divisions, forming out diploid megaspores, and thereby diploid megaspore gives rise to diploid embryo sac and diploid embryo. Likewise, diploid embryo is formed from recurrent agamospermy. Clear, right? This is about two types, adventive embryony and recurrent agamospermy. This is how apomixis phenomena is done, and as a result of apomixis, apomictic seeds are formed which increases the hybrid vigor and desirable traits even without the fertilization. Okay. So, apomixis is a concept which mimics the sexual reproduction. It is asexual reproduction which mimics sexual reproduction. Clear, right? And then polyembryony. Polyembryony is the concept which is first explained by Leeuwenhoek in citrus plants, wherein because of sporophytic budding, okay, because of sporophytic budding, what happens is multiple embryos arise from integuments and from nucellus as a result of budding, sporophytic budding. Multiple embryos, they arise like buds from integumentary tissues and nucellus tissue. Okay, one normal embryo will be there, along with the normal embryo multiple embryos will be present in a single ovule, that is known as polyembryonic. So, with this special modes of reproduction are also done, apomixis and polyembryonic. With this, the complete reproductive biology is exercised in detail and it is briefed to you. In our next class, we will solve the complete objective session on the reproductive biology. After this chapter, we will move on to taxonomy of angiosperms.